Hello friends, I am here with my channel Solving Questions with Gauri ma'am. Today let's have one question from GATE examination for Life Science. Year is 2018. Which one of the following amino acids is responsible for the intrinsic fluorescence of protein? So options are that is proline, or it is a methionine, or it is histidine, or tryptophan is responsible for the intrinsic fluorescence of protein. So to solve this question, we must have idea about the structures of amino acid. So let's go through the structure of each amino acid to find the answer for the question. Yes, starting with the proline, first option. The three-letter code for proline is PRO, and with single letter capital P we denote proline. If we see the structure of amino acid, its aliphatic ring has been cyclized and it is joined with the alpha carbon atom which is present in a structure. So due to this cyclization, this amino acid is known as amino acid. This cyclization creates kinks in the secondary structure of the protein. That's why proline is famous as a helix breaker. Due to this cyclization, proline creates steric hindrance, and that's why it doesn't allow helix to form. So we call it as a helix breaker. This is another speciality of proline when we go for the Nin-Hadrin reaction. In Nin-Hadrin reaction, all other amino acids shows blue color, while in case of proline, proline shows yellow color. So this is again due to the formation of this, or we can say cyclization of this aliphatic structure. This is a cyclic derivative of glutamic acid. The second option is methionine. Three-letter code is MET and single letter is capital M. So if we see the structure, we find that there is a presence of sulfur in the side chain of the methionine. This is having ether linkage in its side chain. This is present in a cereals. S-adenosyl methionine, which is formed from methionine, this is a precursor for the plant hormone ethylene. So this is about methionine. The next option is histidine. Here the three-letter code is HIS, and with capital H we denote histidine. So histidine is having imidazolium ring in its side chain. This is the imidazolium ring present in the histidine. Due to this presence of imidazolium ring, histidine is having pKa values 6. 6 is nearer to the pH of the blood, that is 7 or 7.2. That's why histidine can perform buffering activity at the physiological pH. So due to this property, histidine is present in a hemoglobin. It is also present in protamines and it is present in histone proteins also. The last option is tryptophan. The three-letter code is TRP, and with single letter capital W we denote tryptophan. Here is the structure of tryptophan. It is having indole ring in its side chain. It's a derivative of indole. This shows absorbance at 280 nanometer. That's why this is responsible for the fluorescence of the protein. 3 amino acids, tyrosine, tryptophan and phenylalanine. These are having aromatic ring in their side chains. and this aromatic ring is responsible for the UV absorption at wavelength 260 to 280. Tryptophan is the amino acid which is a precursor for auxine, niacin and serotonin. So here we have found that tryptophan is the amino acid which is having aromatic ring in its side chain, and that is responsible for the intrinsic fluorescence of a protein. If the concentration of tryptophan, tyrosine and phenylalanine is higher in a protein, then that protein is showing absorbance in the UV light. So the answer becomes option D. Tryptophan is the correct answer for this question. So here we have concluded with this question. I hope you like my videos. I request you to please like, share and subscribe for my channel, and also to hit the bell icon. Thanks for watching.